You and Betty and the Nancys and Bills and Joes and Janes will find in the study of science a richer, more rewarding life.
Hey, welcome to Inquiring Minds. I'm Indre Viskontas. This is a podcast that explores the space where science and society collide. We want to find out what's true, what's left to discover, and why it matters. On this podcast, I like to think that we're helping each other make decisions that impact our society and that these decisions are grounded in science, good science. But sometimes science that we think is good science is actually built on a false foundation. Many scientific disciplines have gone through a reckoning over the past couple of decades where studies have failed to replicate, established findings have been overturned. That shouldn't shake our confidence in science as a tool to get us closer to the truth because it's actually doing its job, right? The scientific method is self-correcting. And the reproducibility crisis has led to even more checks and balances. And that ensures that we get even closer to the facts, the real facts. But this story that we're going to talk about today is in some ways a whole other level. We're going to delve into the foundation of the way science is conducted, especially in the US. It is going to be fairly US-centric. And just the entire field and how little mistakes early on could lead to disastrous consequences. And those disastrous consequences have been many millions of dollars, if not billions of dollars, invested in therapies that have proven to be unsuccessful, at least thus far. So I'm not going to tell you much more about exactly what we're going to be talking about because we'll get to that in a minute. But first, I'd like to introduce the person that will be telling the story. Charles Piller is an investigative journalist for Science magazine, and his work has appeared in the Los Angeles Times, the New York Times, and many other great institutions of reporting. He's been honored with many National Journalism Awards and is the author of Gene Wars, The Fail-Safe Society, and most recently, the book that we're going to talk about, Doctored: Fraud, Arrogance, and Tragedy in the Quest to Cure Alzheimer's. Welcome, Charles.
Thanks so much for having me, Indre.
You've had a long career in science journalism, and you're not one of these journalists that just reports the findings without adding all these other layers and elements and really getting at the foundation of how science works. How is it that you became interested in this approach, and what is it that drives you as a reporter?
Well, I consider myself an investigative reporter who writes about scientific issues rather than a science writer who does investigations. It's kind of a way of reminding myself that the techniques, the processes, and the desire to examine things more closely and to present evidence that may be hiding under the surface is far from unique in the world of science, and there's a lot of different ways to pursue it. So I come out of that background, but I've always been very interested in helping distill large volumes of highly technical information into a way that a more general audience can understand it and see layers of truth underneath.
And over the course of your, investigations of your career, I wanted to hear from you how you conceptualize how science is conducted today, particularly in very large, well-funded labs, because that's going to be important as we talk about what's happened here in the Quest to Cure Alzheimer's. How do you think academic research is supposed to function in terms of the role of a principal investigator and all of the people that are working with him and her?
I like that question a lot, Indre. So I think people who have been in science recently know that science is a team sport, and the complexity, the expense, the elaborate equipment that are often essential to doing experiments in a lot of different fields, but particularly in biomedical science, which is an area that I tend to cover the most, This requires many hands. And as you say, a well-funded lab will usually have a bunch of people working on one experiment. Then there might also be collaborators at other institutions. So coordinating and sharing information and being open with each other about how that process works internally in your lab is critical. I guess if you don't mind, I could give one anecdote about what some of the challenges are sometimes. Principal investigators are like the managers and leaders of a lab, often the conceptualizers of experiments, who don't as often do the real hands-on nitty-gritty experimental work. They might be doing it, but it's often trainees or junior scientists who are involved in doing that. And what I've found in my examination of how this has worked the best and the worst is that often when you have principal investigators who are unconventional in the sense that they prevent other people from seeing what they're doing, or they insist on doing certain kinds of things like creating figures or images for papers all by themselves without feedback or input from others, that's usually a danger sign that something needs to be examined more closely.
And I definitely want to talk about the role that images play in terms of, there are certain fields of science where the image is the data, right? And so that's kind of where a lot of this fraud has been kind of discovered in terms of like doctoring, not actual data sets, but the images that are the results themselves. But before we get there, I want to learn a little bit more about PubPeer and what is its purpose. How does it relate to like sort of standard peer review? And the reason that I'm bringing this up is that we've sort of talked about, so if we have a PI and it's a well-funded lab, and as you mentioned, maybe they're not doing all of the bench work all the way down, and yet there is a pressure to publish both for the PI and everybody who's working in the lab who want to start out their own careers. And traditionally, there has been this peer review process, and at Science, of course, is a great example of a very high impact journal that has a pretty rigorous peer review process, and those are parts of the checks and balances to make sure that the findings really are coming from the data, that they make sense in terms of what the person did, et cetera. And yet, that model has incentivized people to, there's all kinds of ways in which fraud has come into peer reviews now and so forth. So what is PubPeer? How are you thinking about the current state of the role that publications play in terms of both how we consume science, but also how individuals who are doing the science are rewarded for the work that they're doing?
Yeah, so you're absolutely right that peer review for scholarly publications is the bedrock of their believability and their ability to shape and improve our scientific understanding. And historically, it's been, ultra important for advancing science and still is. However, there are some weaknesses within the peer review system. And I'm going to get to your question directly about PubPeer in a second, but just for context, when peer reviewers are asked to review papers, they are not trained specifically in looking for potential fraud or misconduct. They don't know how to evaluate whether images, for example, have been purposefully and falsely manipulated, perhaps to accentuate their support for an experimental premise, or maybe just to completely change the results of the experiment in order to make it look successful when really it came out with data that wasn't supporting the premise. So because peer reviewers are not trained to do this, they're not looking for it. They're not expected to do it. That responsibility should and normally does fall to the editors of the journals. So there's a couple layers of quality control. There's the peer review process, and then there's the journal editors themselves who are looking at this and, as you say, trying to make sure it makes sense, it's in the style of the publication, that the experiment is significant enough to publish, things like that. And unfortunately, historically, they also have not been examining images in that way with an eye towards the possibility that their colleagues in academia or in industry who are writing these papers are not trustworthy, just to put it bluntly. And so what happened is that over a period of years, there was a kind of set of concerns that arose that images were not being examined closely. And the site that you mentioned, PubPeer, is an online, it started as a so-called journal club. So it was an informal way for people to post questions about articles and to post questions about methodology or results and try to have a dialogue with the authors of those scholarly articles. And what it evolved into in recent years is really predominantly a way for people who are challenging or questioning the veracity of scientific images to point those out and to ask questions of the people who produce those images and say, look, this doesn't look quite right to me. Can you respond by explaining why perhaps I'm mistaken? Or can you show evidence to suggest that this image is true and correct? And normally this is done anonymously. So there's a kind of a protection for people who are junior scientists to, or even non-scientists, to be able to ask questions and not feel that they might have their careers damaged by going after people's papers who maybe are higher up than they are in the food chain of science. And so this process became very popular, and they're kind of developed a kind of small army of people who call themselves image sleuths, who may not even have scientific training, but have an expert eye for how scientific images are supposed to look and how they might be altered digitally. And then they can use that, to ask questions of the authors. So it's controversial. Many people in science object to this process and object to it being done in the way it is, particularly the anonymity of the process for the people asking the questions, or at times even making accusations that something improper might have been done. But I think it gets directly to a huge problem in the scientific literature right now, is that journals are not taking enough responsibility to do a rigorous, thorough look at the veracity of these scientific images the way they do for the sense of the experiment, the methodology, the reputation of the researchers involved. They check all of those things very well, but by and large, they're way behind in certifying the believability of the images. And so outsiders have taken responsibility for it, for better or worse.
A lot of in the reproducibility crisis, one of the checks and balances that people have asked for is open, that you have to literally submit your data set to an open science repository. That's a requirement for publication now for a number of journals, so that anyone can come in and look at your data and reanalyze it and make sure that they're coming up with the same result that you came up with and that you reported. So that's a check against essentially just creating a fraudulent data set. But when it comes to images, that's more challenging. And so maybe we should help our listeners understand the role that these kinds of images as data play, and let's use Alzheimer's disease as the sort of main kind of area here, since that's the focus of your book and where a lot of this fraud that you describe has come from. So how can images be data? How is that something that is used as proof of the science, and in particular in relation to Alzheimer's disease?
Yeah, I can give you two really concrete examples. So two kinds of images that are very common in laboratory research associated with Alzheimer's are micrographs of brain tissue. So these are highly enlarged slices of brain tissue. And the other is something called Western blots. So to take the first, micrographs, so often in experiments, scientists are trying to demonstrate that a chemical or a antibody or some treatment of tissue, usually in mice for a lot of the experiments, but also in people in some cases, has a effect that's observable in these highly enlarged tissues samples. And if those samples are manipulated in ways that resemble a real picture, but move certain portions of them around to accentuate the apparent effect, say, of a compound using to see if it has a reaction in the tissue, then you can suggest that the experiment was very successful in demonstrating that this particular compound has promise as maybe something you could develop into a drug for people someday. So that's one example. Western blots are a really different looking. I think people can easily visualize a microscopic tissue sample picture, but Western blots are different. What they are a technique for displaying the proteins, various kinds of proteins and their relative amounts in a tissue sample or in a blood sample. And there's a kind of an elaborate electrical process where they're put into a kind of plastic frame, and electricity kind of forces the sample through. It's kind of like a sieve, and the heavier proteins stay up at the top of the frame, and the lighter proteins, the lower molecular weight proteins, kind of filter down to the bottom. And what you end up with is a photographic process that takes a picture of your results. And the results show which proteins are present in the sample and in what amounts. And so why do we care about this in Alzheimer's disease? Well, Alzheimer's disease is thought by many scientists to be a disease that's caused by a particular kind of protein called amyloid beta. And there are many forms of amyloid beta, different molecular weights. And so they use this test to see how much of a particular kind of protein is in the sample and what effects do treating the sample with a particular compound have on that protein. So it's a way of really understanding at a very deep level what you have when you, say, take a piece of brain tissue from a mouse and grind it up and put it through the system and see what's there. And it can be used to understand many concepts in how to move forward in developing therapeutics.
You know, at the sort of height of the amyloid hypothesis, I guess I think of it as sort of maybe 20 years ago, there was this real excitement that we found the molecular basis of Alzheimer's disease. And now all we have to do is find the pharmaceutical therapy that will knock it out, get rid of it, clean it out, whatever it is that we decide is the best way of limiting its effect. Is essentially a misfolded protein that's gathering up in the brain and causing all of these issues. And 10 years later, 20 years later, it's a great disappointment that we don't have these kinds of drugs. And I think in reading your book, it started to, I knew that like the amyloid hypothesis was losing, steam, that there were other issues. One of the things that you brought up both in your book and in a recent New York Times article is that there's lots of evidence of people who have a lot of the same, amyloid in their brain, and they don't have symptoms of Alzheimer's disease. So the story there, was like, well, they must have all these other resources, cognitive resources that they're drawing on that allow them to protect themselves from these cognitive symptoms. So, good lifestyle, lots of social interactions, highly active brains. I mean, it launched a whole field of brain training games, et cetera, protect you. Even if you have the wrong genes, you can still protect, from this disease. And it wasn't until I started reading how you were describing these like little tiny missteps seemingly that individuals make that start to blow up. And it's almost like a butterfly effect in my mind of like it then, because of the way science is conducted, because of the way funding happens, you've got these early things, maybe almost innocuous little manipulations of an image that then blow up into many, many, many millions of dollars wasted. How do you think about sort of the role, and is it just a single butterfly wing that flapped and caused this? Is it a whole series of things. And I guess one of the reasons I'm asking is that I don't fundamentally believe that people that maybe altered these images, that all of them anyway, intended to defraud. I still hope and believe that they thought that what they were doing was going to get us closer to a pharmaceutical therapy.
Sure. Well, obviously, it's very hard to know someone's exact motives, but I think you're right. I would say, first of all, I just want to say that even though I wrote a book that is largely about falsification of research, I believe deeply in the fact that most scientists are people of integrity and are very concerned with moving human understanding forward in the most practical and efficient way, and are committed to doing these experiments honestly. That said, it may not take an enormous number of people who are, you might say, cutting corners, or you might say committing fraud, depending on the severity of the circumstances, to skew thinking in the field. So let me take the example of the amyloid hypothesis that I think you articulated well to explain why your idea about that might hold a lot of water in regard to understanding this problem. So back in the 90s and early 2000s, the amyloid hypothesis had been essentially become the dominant way of thinking about Alzheimer's disease. And I would argue that it is still the dominant way, although there are many other ideas now that are slowly beginning to gain traction. And I think for people who want to see a variety of ideas explored for their potential, they should feel heartened by that. Now, that said, back in the early 90s, there were already starting to be a bunch of failures of the amyloid hypothesis in regard to therapies being developed based on it. So this idea was, what you do is you create a drug or an antibody that would go in and flush the amyloid proteins, these sticky plaques and these also soluble forms of amyloid protein, out of the brain. And then, in theory, it would arrest or possibly even reverse the symptoms of Alzheimer's disease, the cognitive decline that we all know is the tragic and terrifying part of that disease. So what happened was that none of these things worked. They were successful in taking the amyloids out, but they were not successful in improving cognition, arresting the decline of Alzheimer's patients cognitively. So something happened, something very important to the hypothesis happened in 2006. And this was a seminal experiment primarily done by professors at the University of Minnesota that found that a particular protein, particular amyloid protein that they named amyloid beta star 56, their star protein, a kind of a catchy name that actually assisted them in gaining mind share, that this protein, when it was extracted from the brains of mice and purified and then injected into rats, could cause symptoms that could be described as akin to the memory loss symptoms that people experience when they have Alzheimer's disease. And so it was the first time that a specific compound, a specific protein, was identified as being directly associated with causing Alzheimer's dementia. And the study was published in the journal Nature, which many of your listeners must know is one of the most prestigious scientific publications in the world. And it was a study that has been cited thousands of times in the literature. It was vastly influential. And it was cited not just by all different kinds of scientists, but notably by the leading figures in Alzheimer's research, some of them dozens of times, in supporting their own ideas about the amyloid hypothesis. So this study actually was, in a sense, the genesis of my book. And because it involved an examination of that paper, the 2006 paper in Nature, this is something that was done by a scientist at Vanderbilt University, a guy by the name of Matthew Schrag, who I worked closely with and was a source of mine for a story in Science about that paper and some related research. And what Schrag found by examining those images very closely in 2021 and 2022, so many years after the paper had been published, was that there was strong signs that they had been edited improperly using a program like Photoshop to make the images appear to suggest that the experiment was successful. And when in reality, the real images that came from it didn't look that same way. And so I wrote a story about this in Science Magazine. It was a pretty controversial story. It really stunned the Alzheimer's research community and got a lot of attention among people in the broader public. And the reason is because it called into question this very important building block of the amyloid hypothesis. Now, let me say, to use your butterfly wing example, I don't think this one study is responsible for the waste of millions, billions of dollars in research funding, because there were a lot of people who believed that this certain form of soluble amyloid protein that was akin to the kind that was found in this experiment did indeed play a role in Alzheimer's disease. And so it wasn't one piece of evidence, but it was an important piece. And in my view, and I think in the view of many scientists who were experts who I interviewed for this story and for my book, what we saw was that NIH funding, and that's the National Institutes of Health, who are the primary funders of biochemical medical research, basic research in this country, they increased funding for this particular kind of research so dramatically that billions of dollars were spent on it in the succeeding years, not just from the NIH, but also from industry, trying to develop drugs and treatments that would be based on that idea. And I can talk about those as well if you're interested.
Yeah, well, so I think that's a really great way of sort of setting up what I wanted to talk about next, which is that then led to the incentive. So yes, maybe there was this one paper, and I'd love to talk more about Matthew Schrag, that's his name, right?
Matthew, yes, Schrag, yes.
Schrag, motivation for actually going back, so many years later and looking at that and doing that kind of careful work. That's really interesting too. It's a great, interesting story, and how it took a long time for him to trust you and the role of whistleblowers, in the field. But, before we get to that, there is this other component of the NIH or these big granting organizations where they can call for proposals and they can devote a lot of money to particular scientific topics that they find have a lot of promise or in line with their own goals, and in that case incentivize more and more people to do work, it's still obviously good science, but there's a kind of nudge towards, finding, kind of confirming that hypothesis rather than disconfirming, or, which is how science should work. So, and like an example I would give is that, the NIH, that is the main funding organization that gives out these massive grants, and a lot of these researchers, their salary depends on getting these grants, and if they're PIs, the salaries of all the people that work for them depend on getting these major grants, and that you have a better chance of getting the grant if you write a grant proposal that is in line with what the reviewers at the NIH think is a good project. And so you can see how there are these little subtle nudges that if the NIH is saying, well, we really want to go after the amyloid hypothesis, there's a lot of motivation for you to say, okay, well, I wasn't 100% sure that was the direction I wanted my lab to go, but it's the direction that's going to get funded. So why don't I write this grant? And, then it goes on and on and on.
Yeah, that's exactly right. And in fact, I think what you're describing is a kind of confirmation bias in the field. When the conventional wisdom is funded and supported and is backed by the institutional authorities of science, and I mean the funders and the journals in particular, then it really becomes very difficult to fight against it. And in the book, I have stories of scientists who did try alternate paths and were shot down or took many years to try to establish themselves because of the pressure of conformity. Now, science is hardly unique in that way. And, I like to say that confirmation bias is also a problem in my business, in the business of journalism. And so what I try to do really hard with, especially with tough problems like this, is not just be skeptical of the topics I'm looking at or the people that I'm investigating, but to be skeptical of my own assumptions, because it's just a healthy way of thinking to try to reevaluate and get to answers that may be unexpected. So, unfortunately, I think the entire field became, I don't know, maybe it's extreme to call it captive to this one idea, but very much this idea was the dominant way of thinking. And I might add that part of what I did in the book is I did a really broad examination of scores of top scientists in the field, looking at their work for possibility of image manipulation that might have skewed thinking in the field. And what I found is that out of the hundreds of studies that were evaluated as part of my work for Doctored, that the large majority were studies that involved the amyloid hypothesis. And that's not surprising. When one idea is so dominant in the field, it becomes easier to fake data because you are meeting the expectations of your audience, the journals, the funders, and the scientific readers.
I think that's a really interesting and important point because, of course, even if you're not, if there's a gray area, let's say, in between, because if you want to publish in Science or Nature, like your figures have to look good. Like we're all told, you do the figures first, and the figures have to not just, I don't mean doctor the data. I mean, they have to look nice. Like you have to put some time into making them aesthetically, clear and interesting and sort of nice to look at. That's really important. And so if you, think, well, if I just kind of remove some of these things that maybe are muddying the picture a little bit and just make my findings really clear the way you would, in a graph, now you do that for some of these sort of more data images. But you can feel pretty confident on that, knowing that you're building this on a foundation of, many, many, many other studies. So maybe it feels like, well, if I just kind of do this here a little bit, it makes the story clearer. And, this is not a novel finding or like an entirely novel direction, right? That this is building on an established set of findings. That it seems like maybe it's easier to convince yourself that's okay.
Yeah, I couldn't agree more. And I think that, again, there's a kind of a continuum of actions. I think people sometimes do this so-called, as you say, beautification process of images to give them a kind of curb appeal for the scientific journals. And then, the next step of that might be, well, I can see that the image shows the veracity of my thinking, but kind of weakly. If I just sharpen this up a little bit here and kind of fuzz that a little bit there, I'm not really changing anything. I'm just making it clearer, something that I believe in. And then the sort of more terrible and final step of that might be, well, I believe deeply in my findings. And even though the data isn't supported by the images don't support the premise of my experiment, I know in my heart that this is good science, is important science. I'm just going to provide an image that displays what I think it should display. And that is, of course, frank misconduct, outright fraud in some cases. And this continuum exists in all walks of life. Scientists are people and no more or less honest than people in other walks of life. So when I say that I've found numerous examples, even from important, illustrious researchers, of doctoring images, I'm not suggesting that this is just the norm in science. It's not. I believe deeply, again, in the integrity of most scientists. And I think it's just a terrible shame that a relatively small number of influential people can steer thinking improperly for their own motives, which might be money, prestige. There can be gigantic financial rewards, even in situations where the data is corrupted.
One of the most moving parts for me of the book was when you traveled to the University of Minnesota to spend some time with Dr. Karen Ash, and you wrote about her perspective. Tell us that story.
Yeah, so Karen Ash was the senior scientist behind this seminal 2006 paper in Nature that I was describing earlier. And she was the mentor of a guy by the name of Sylvain Lesné, who was at the time a postdoc in her lab. And the two of them were behind this discovery of amyloid beta star 56. And I had been trying to interview Karen Ash for literally for years after I first learned about this situation, but she always refused to talk to me. Finally, she consented, and I flew to Minneapolis to spend a couple days in her lab. It was it was amazingly cold there, I might add. It was during the dead of winter, and the wind chill factor was 20 below zero when I stepped off the plane. But I was welcomed into her lab, and then I had I thought, a very strange experience with her. First of all, I want to say that Karen Ash herself, I don't regard her as someone manipulating data. I don't see any evidence that she personally changed images to support an improper or false premise of an experiment. But she was the boss there, and it was pretty clear that she saw these images, did not detect problems, did not look further, took credit for the experiment, had gigantic career advancement associated with it. And so say what you will about her culpability, if you want to use that term, in the fact that the images were incorrect. But I think she was someone who deeply believed in good science nonetheless. She was bitterly angry at me for having exposed these problems in her lab and in her experiments and that of her younger colleague, Lesné. And she basically took me to task for it for a couple of days. And it was a very unusual kind of interview. I respect Karen Ash, but I think she represented someone who basically maybe to me wasn't practicing the same degree of self questioning, the kind of being skeptical of even of your own ideas that I think is healthful in a productive and thoughtful scientist. And as a result, she maybe was put in a bad direction by the apparent falsification of images by this guy Lesné. There's never been any conclusive investigation completed in that case, but the paper was retracted, and all the authors except Lesné, who were consulted, admitted that the images apparently were falsified. So it was a strange experience, and also Karen Ash is a deeply religious person, and I want to say that many great scientists are also have deep religious faith, and there's no contradiction there in my way of thinking. That said, I think she applied a strongly religious kind of judgmental attitude towards me and felt that I had transgressed the precepts of the Bible in criticizing her and her work in the way that I did in my article, which analyzed that experiment. It was a little bit of a strange experience, I have to say.
And of course, she wasn't. This was one paper, but there are other people that you talk about in the book, like I think Maslia is how Dr. Maslia is how I pronounce it. Where there are like 800 papers that they're not all fraudulent, of course, but that there's like these large numbers too of other papers, and in some ways I wonder how you approach that kind of a conversation with someone who maybe who it's not just one particular paper that you're pointing to, but rather perhaps a tendency to overlook or not supervise as closely, the people that are allowing them to put their names, they're writing the papers and then they put their names on it. And I guess the question here is, do you see a difference in terms of that response when you're, when it's one paper, it's one thing, but when you're kind of questioning someone's larger body of work, do you get a different reaction? And also sort of what you think about what are the lessons that we should learn about, like when you hear somebody has 800 published papers, that sounds incredibly impressive. But, for me as a scientist, someone who works in the trenches, it also sounds totally impossible in terms of the care that I would want to give to the papers that I put my name on it. And so this question of authorship for these big team projects and, who is responsible, did you find that there was a difference in how people reacted to your reporting, depending on, I guess, how they, how loose they are with adding their name to a paper, for example, versus, yeah.
Sure. Yeah, I get the question. Thank you. I think it's a really interesting question because the reactions are very different. So in the case of Karen Ash and Sylvain Lesne, a big part of his body of work was similarly implicated in apparent image manipulation. He would never talk to me. He's never responded to any of my requests for interviews. In the case of Maslia, so this is a very interesting case because this is a guy who in 2016 was hired as the director of the Division of Neuroscience for the National Institute on Aging. And this is the agency of the government that is the primary funder of basic research in Alzheimer's disease and some clinical research as well. And so Maslia was an illustrious researcher before even coming to NIA in 2016. He had, as you say, 800 published papers. So in his case, there were 132 papers that were found by a team of image sleuths who worked with me on the book. And these 132 papers all showed signs of either manipulation of images improperly or reuse of images as if they were new images in separate papers, sometimes published years apart, another kind of misuse of images. And the thing about Maslia is that he was in this important role at the National Institute of Aging. And when I approached him with the evidence of apparent misconduct going back literally decades, he wouldn't speak to me. And this is the standard experience that I've had. I've approached scientists on these matters dozens of times in preparation for writing Doctored and in my articles for Science magazine. And when people have no story to tell that seems like it would exonerate them from apparent misconduct, they won't talk to me. It happens over and over again. And so it's discouraging because my approach, my technique as a journalist is that there are never any surprises. When I am writing about a person's body of work, they know everything I know when I approach them. They receive a copy of the dossiers that are prepared that show every single paper and every single question about every single paper. They get a long list of questions from me about every aspect of the story so that there's no reason for them to ever come back to me and say, well, if I had known you were going to ask about that, of course I would have spoken with you, because that will never happen in a story that I'm reporting. That said, they don't talk to me because they don't have a good story to tell. It's too difficult for them. Let me just say one more thing about Maslia, if I may. Again, even though most scientists are honest, having a guy at the pinnacle of this pyramid of funding who was one of the most important people for setting policy and direction, for studying neurological disorders, not just Alzheimer's, but also Parkinson's and others. For him to be the guy at that post with his record of apparent falsification of data, it's very sobering. And it shows that the institutional authorities of science, not just the journals that didn't catch these problems before they were published, but the funder, NIH, who didn't do their own due diligence to try to examine whether the person they were hiring for such an important job might have a suspect past. So what we need is for a higher degree of vigilance by the institutional authorities of science. And what I mean by that are the journals, the funders, and the regulators like FDA. They need to take seriously that image manipulation, scientific misconduct, is not a super rare thing. They need to make it a part of their routine and to try to catch these things before they pollute the scientific literature. Now, that said, I know that there's a real force going on in Washington, D.C. with the new administration. And there are many people who are going to be in positions of power who, frankly, distrust a lot of science, even beyond the possibility that some minority of scientists may have improperly acted with regard to their data. And I just want to say that, excuse me, I just want to say that personally, I think it would be a terrible mistake to tear down these agencies and to consider them to be somehow terrible at what they're doing because there are some bad things that need to be corrected. They need to be supported, strengthened, and actually given better funding to be able to do their jobs better, including the careful analysis of the possibility that people are doctoring work that gets funded or gets published.
So on that note, I want to remind our listeners that Charles Pillar's book, Doctored, Fraud, Arrogance, and Tragedy in the Quest to Cure Alzheimer's is now available at booksellers everywhere. And I think it might, not just help us understand when science goes wrong and how it can go so wrong, but also why we don't have a cure for Alzheimer's yet, despite all the promise and all the funding. And so what do you think are the next steps for somebody who is interested in Alzheimer's research or who, whether they're, a potential funder or a scientist, like where do we go from here so that we can get closer to the cure, given that a lot of the foundation, in some ways has to be questioned?
Yeah, I would like to leave listeners with a little bit of an optimistic note because I think because of the coverage of the concerns about the amyloid hypothesis and because of the, at best, very weak results of some of the drugs that have been developed to treat Alzheimer's based on that idea, there have been increasing interest in looking at alternative theories about the disease. And because of that, there has been slowly increasing funding and some, I think, very encouraging, perhaps optimistic possibilities coming out of it. Let me give you a couple of examples. One is there's fascinating science going on around the idea that infections, viruses perhaps that are latent within the brain, might be instrumental in causing or influencing the course of Alzheimer's. One of these is herpes virus. And in fact, there's clinical trials going on right now to examine the role that might play in the disease. So that's number one. Number two, GLP-1 inhibitors. These are, of course, the class of drugs that have become the kind of almost magical weight loss solution for so many people and have been talked about and begin to be applied for other kinds of conditions such as diabetes. And they're being talked about as being kind of a miracle drug. Believe it or not, they also have been tied to the possibility that they could be effective in Alzheimer's disease by reducing inflammation, which many people believe could be a key element of how Alzheimer's develops and how it progresses. So there is a gigantic clinical study being done right now involving GLP-1 inhibitors to look at its effects on early Alzheimer's. And those results should be in sometime in 2026. So I'm very much looking forward to hearing what those results are. The other thing I wanted to say, and there's a lot of snake oil being sold about Alzheimer's remedies and brain health and supplements and things like that. Some of those are patently false ideas, and some I just don't know anything about and I can't really comment on. But what I can say is that each of us has some agency in our own health and as it regards to Alzheimer's disease. Healthful habits, good dietary habits, watching your cholesterol, watching your blood pressure. These are all important things to do to reduce your chances of especially getting Alzheimer's at a more early age, and also of perhaps forestalling the worst effects of Alzheimer's should you be unlucky and get it at an older age. Plus, let's face it, there's great benefits to be had by being healthful and being active and exercising for all of us, not just for Alzheimer's, but just for our general health. So I would just say to people, it's a scary disease. It's a discouraging set of affairs when you have a lot of scientists who have unfortunately put their own careers or their own financial prospects ahead of truthfulness and have faked research in this field. Very discouraging. On the other hand, there are all reasons for hope, and there are reasons for us to be able to take responsibility to some degree in improving our own health and safeguarding ourselves.
Well, Charles Piller, thank you so much for being on Inquiring Minds.
Indre, I've really enjoyed talking to you. Thank you again.
So that's it for another episode. Thanks for listening. And if you want to hear more, don't forget to subscribe. And if you'd like to get an ad-free version of this show, consider supporting us at patreon.com/inquiringminds. I want to especially thank David Noel, Herring Cheng, Yushi Lin, Jay Henry, Joelle, Dale Lemaster, Charles Blyle, and Matthew Rowland. Inquiring Minds is produced by Why Not Alchemy. I'm your host, Indre Viskontas. See you next time.